Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic. Today our topic will be based upon an important question that had been asked to me from one of my students, that asked to me, what is the difference of the DNA molecule from ideal crystals? What is the experimental diffraction pattern of DNA molecule? Explain why it looks that way. It is an important question. I will try my best to answer this question. Firstly, we will have to know that what is DNA? The answer is that DNA is deoxyribonucleic acid that is present in the nucleus of a cell. This cell can be animal or plant cell. So we can say that DNA is present in both plant and animal cells. DNA is main functional part of a cell, like the center of a cell, which performs two major functions. First one is that DNA directs many chemical reactions in the cell by transcribing genetic information from DNA to RNA, meaning that suppose it is a big view of this smaller nucleus. In its big hollow space, we can easily understand the next reactions. Messenger RNA is synthesized from DNA by transcription process. Then by using this messenger RNA, ribosomal RNA and transfer RNA. So all these three RNAs collectively take part in translation process and these synthesize polypeptide chain. Finally, this polypeptide chain is highly organized to form actual final protein. Second function of DNA is that it stores genetic information in the form of genes. These genes are responsible for the specific characteristics of an organism's body. The genes for eye color determine that what will be the eye color of a particular organism, which can be a human or an animal. It could be black, brown, green or some other mixed color like honey brown. Similarly, traits for hair color, skin color, body shape, the size of the fingers, thumb design, till every trait of the every organism is controlled by genes that is present on the chromosomes, and these chromosomes are made up by the DNA during cell division. As these genes are made up by DNA, that is why they take instructions from DNA, and then according to these informations, these genes perform all of the functions, all the traits of a living organism. So DNA transfers the genetic information from one cell to next cell, meaning that from parental cell to daughter cell, and also from one generation to next generation during cell division. As DNA is present in a nucleus that consists of some special parts such as nuclear envelope, nuclear pores, nucleoplasm and nucleolus. We will study all these parts in detail in our upcoming videos. The DNA changes to chromosome in the dividing cells of eukaryotes, meaning that only during cell division, whereas in non-dividing cells it remains in the form of chromatin material. Resultantly, we can say that DNA controls all the cellular activities of a cell of any organism. DNA is a self-replicating organelle. It consists of a large number of subunits that are repeating units of the DNA molecule known as nucleotides. Very huge amounts of nucleotides are assembled to each other to form long chains of DNA. To understand the nucleotide concept and DNA structure in a premium way, you must watch my videos about nucleotide and DNA as a genetic material, because from these videos you can get minor details about DNA structure. On the other hand, crystals are obtained naturally from various different sources such as rocks, oceans, etc. But these are not present in a living cell in very huge amount. A crystal can be defined as the solids in which atoms, molecules or ions are arranged in a definite three-dimensional arrangement. Its examples are sodium chloride, potassium nitrate. The crystals show many properties such as geometric shapes, melting points, cleavage plane, anisotropy, polymorphism and allotropy. Crystals can also be shown transition temperature. All these properties would be according to the crystal nature. Crystals can be divided into two forms. First one is ideal crystal. A crystal in which atoms are arranged in following certain symmetrical parts in throughout the entire crystal without any impurity is known as ideal crystal. So ideal crystal is an infinite single crystal with 100% order and purity. Such a crystal is ultimately known as a perfect crystal, that is also called ideal crystal. But ideal crystals neither occur in nature nor can be artificially prepared by any artificial method. In practice, crystals will have some of the other types of deviation from the ideal behavior. So a crystal having some of the other type of deviation from the ideal behavior are called as real crystals. That is second form of crystal. Real crystals are never perfect and have some type of violation of long range order. There will be some impurity atoms placed at some lattice point of that crystal. Any deviation from perfect periodic arrangement of atoms or molecules is stated as imperfections, and real crystals will always have some of the other types of imperfections. Next, we will see the experimental diffraction pattern of DNA molecule. Who performed it, how performed it, and why performed this X-ray diffraction pattern of DNA molecule. Students, X-ray diffraction pattern brought great revolution in molecular genetics. But before going to the detail, I want to present tribute to Rosalind Franklin by telling you about her. Rosalind Else Franklin was a woman, an English chemist and X-ray crystallographer. The lifetime of Rosalind Else Franklin was from 25th July 1920 to 16 April 1958. Her works on the molecular structure of coal and viruses were appreciated in her lifetime, but her work on and contributions of discovery of the structure of DNA molecule was mainly recognized after her death. Rosalind Franklin obtained X-ray diffraction photograph of DNA by passing X-rays through crystals of DNA. That were her photograph 51. This photograph suggested the double helical structure of DNA. With the help of this photograph and Chargaff's rule, which stated that the amount of adenine in DNA is always equal to the amount of thymine and the amount of guanine always equals to the amount of cytosine. So there is always equal proportion of purines and pyrimidines in nucleic acids. James Watson and Francis Crick designed DNA model in 1953. They got Nobel Prize for this model in 1962 along with Maurice Wilkins. Rosalind Franklin died on 16 April 1958 due to ovarian cancer. Exposure to X-rays is considered to be a possible factor in her illness. She has played an important role in the discovery of DNA structure. Now come back to our experimental detail. Rosalind Franklin's photographic technique relied on the wave property diffraction. Today, diffraction is an important and common topic experiment in physics classes. Franklin used X-rays that have very shorter wavelength to image something much smaller. With the diffraction pattern, any object can be easily analyzed and used to determine the original object, still it be a solid graphite or DNA. X-ray diffraction pattern has been used to image and analyze any object more clearly. When the X-ray beam that is bombarded from the X-ray source hit the DNA molecule, keep in mind that DNA molecule was in the form of crystals. So after hitting the X-rays to that crystal DNA, those X-rays were diffracted into a pattern. From this pattern, the molecular shape can be determined. Why did she have to use X-rays? Reason is that the wavelength of the X-ray is so much shorter, whereas the wavelength of the visible light is larger. Because of this, we can make the images of the object rather than much smaller. It is the image of 51 photograph of Rosalind Franklin's X-ray diffraction pattern. Rosalind Franklin prepared this X-ray diffraction pattern of DNA in the laboratory of British biochemist Maurice Wilkins, who prepared DNA fibers for her. She obtained many different photographs, out of which 51 photograph is most important. Her X-ray diffraction pattern suggested that DNA molecule had a shape shape of a helix like this. That is the first point. So this is the X-ray crystallographic diffraction pattern of photograph number 51 of DNA, accomplished by Rosalind Franklin and Rosalind in 1952. That is known as B form. Both scientists James Watson and Francis Crick were struck by the simplicity and symmetry of this pattern. The ascertained 2x in the X-ray photo is the signalized pattern of a helix. Because the X-ray pattern is so regular, the dimensions of the helix must also be the consistent. For example, the diameter of the helix remains the same, that is of about 2 nanometer. That is the second notable point of X-ray diffraction pattern of Rosalind Franklin. Using this pattern, Rosalind Franklin and Maurice Wilkins were capable to derive some basic dimensions for the DNA structure. In this X-ray diffraction pattern, closer the spots, the larger the actual distance. The horizontal bars actually corresponds the helical turns. The vertical distance between the bars is 34 angstrom. That is the measure of the height of one turn. Distance from the middle of the X-ray pattern, the top is measured about 3.4 angstrom. This corresponds to the distance between two stacked base pairs. Since the height of one helical repeat is 34 angstrom and the distance between two stacked base pair is 3.4 angstrom, there must be tiny nucleotides for helical repeat. The pitch of helix for the degree of rise can be calculated from the angle the X makes with the horizontal axis, like distorting the helix. Now you have get an idea that how the helical pitches are related to X-ray diffraction pattern. From this X-ray diffraction pattern, Franklin recommended that the phosphate groups were on the outside. From the analysis and measurements made by the Rosalind Franklin, third point is that a complete helical turn every 3.4 nanometer. These basic dimensions of the DNA structure were known by Rosalind Franklin. I hope you are satisfied with my efforts. How you find this lecture? By liking it and by subscribing our channel. At the end, I will advise you, please share this lecture to your those friends that are fond of science knowledge. Say the prayers and pray for us and yourself. Thank you for watching this your video.